So we're going to look at the introduction to animal diversity, which is basically, first part is going to be animal development, and then we're going to look specifically at invertebrates. We are going to be in the domain of Eukarya, the kingdom of Animalia, the phylum are all these that we're going to be looking at are, hang on, let me get this going. We're going to be looking at Porifera, Cnidarians, Platyhelminths, Nematoda, Mollusca, Annelids, Echinodermata, arthropods. Now, all of these, as you notice, these are all going to be invertebrates. Our vertebrates are going to be the Chordata phylum, and those are all going to have a backbone or a spinal cord. Now, the evolution of animals. So it says that all animals are going to be multicellular, heterotrophic. They're going to reproduce sexually, most of them. Some of them reproduce asexually. There's two branches that we're looking at, invertebrates and vertebrates. And in this video, we're going to look at invertebrates only. So the ancestry of animals. The first thing that we need to write down in our notes is going to be that you need to know this hypothesis, the colonial flagellate hypothesis. And so basically that states that animals descended from an ancestor resembling a hollow spherical colony of flagellated cells like echinodermata. And a echinodermata, what that means, if we look at one right over here, it means that it is basically a collared-looking cell right here, a collared cell, and with the flagella. Now it says the transition from colonial flagellates to multicellular animals may have begun by aggregation of a few flagellated cells. And so basically this hypothesis is attractive because of its implications regarding animal symmetry. And so we're looking at actually like at sponges where there's two tissue layers. They may have arisen by infolding of certain cells into a hollow sphere. So all diversified body plans of animals were present during the Cambrian period. So this needs to be noted. Then it looks at, if we look at the symmetry of different, there's basically three types of symmetry in the animal kingdom. So the first that we need to note is asymmetry, where there is no particular body shape. For example, a sponge. Then the next is radial symmetry, where you have two identical halves, for example, like a jellyfish right here. So it has this central axis around it. So no matter which direction you cut it, it's all, each side is going to mirror the others. Then what happens is this enables an animal to reach out in all different directions from one center. Then the third is going to be bilateral symmetry, where you have a definite right and a left half. And so like here our example is a turtle, or it could be a human being. So I would definitely note these three types of symmetry. We're going to watch this video over Hox genes, and this will kind of describe it, and then we're going to put a few notes in.
Hox genes and the evolution of animal body plans. Animals have a variety of body plans. Flies have 6 legs, mice have 4, and millipedes have many. Scientists have proposed that changes in a group of similar genes called the Hox genes have created the diverse animal body plans we have today. To test this hypothesis, our lab has focused on crustaceans. Crustaceans are an excellent group of animals for studying body plan evolution because they have many different body plans. By examining Hox genes in crustaceans, we can understand how these genes have changed to produce new body plans. In particular, our lab focuses on the sandhopper. Let's stretch out the sandhopper body plan so we can see each segment. The sandhopper has antennae, many mouthparts, claws, forward walking legs, reverse walking legs, swimming legs, and anchoring legs. First, we figured out where each of the Hox genes is turned on. Then we deleted each of these Hox genes to see how this would affect the animal's body plan. For example, if you delete the Hox gene called abdominal A, the animal's backwards walking legs turn into forward walking legs, and the animal's swimming legs turn into anchoring legs. This means that abdominal A is necessary for making backwards walking legs and swimming legs. The portion of the body that turns on the abdominal A gene has changed over evolutionary time. If we look at other types of crustaceans, such as shrimp and pillbugs, we can see that the part of the body that turns on abdominal A has changed. This change leads to a difference in the body plan of these other crustaceans. Currently, we are starting to determine what is responsible for the changes in where abdominal A is turned on. By figuring out how this change happened, we can develop a better picture of how the Hox genes have changed over evolutionary time to produce new body plans.
So if we look at this image right here, we are like comparing, this says Hox genes are highly conserved genes encoding transcription factors that determine the course of embryonic development in animals. In vertebrates, the genes have been duplicated into 4 different clusters on different chromosomes, Hox A, B, C, and D. And so genes within these clusters are going to be expressed in certain body segments at certain stages of development. So shown here, we're looking at the homology between Hox genes in mice and humans. Now, note how they are indicated. Each one is indicated in a different color. So 1 through 4 are going to be this pink color, 5 through 6 are this color, and then so on, and 9 through 13 are green. So it says if we're comparing these right here, it says that each segment has where those genes are identified at, whether it is the thorax region or the abdomen or whatever region it's color coded, and you can note them with these genes. So what this is asking us over here, and I would probably write this down because I think this might be on the quiz, says if HOX13 gene in a mouse, which 13 is going to be the green color, so it's going to be here on like kind of the tail region. If it was replaced with a HOX1 gene, how might this alter animal development? So if it was replaced with 1, so 1 we note like on a human is the head, on the mouse it is a head. So if we replace this gene right here, what is going to happen with this mouse embryo? How will it alter the development? Could it possibly have 2 heads now and no tail? Okay, looking at embryonic development, there's a few of these terms that we're going to have to look at. When we're looking at the first tissue layers that appear, we note those as germ layers. And these all give rise to different organs and organ systems. If something is diploblastic, means it has 2 tissue layers, and triplo means 3. And then we're going to look at what a protostome and a deuterostome is.
The embryonic development of animals always begins with the union of an egg and a sperm. When they join together, then the DNA combines into a zygote, and then mitotic division begins. When mitosis continues during cellular division, then it creates a blastocyst, which eventually becomes an embryo, and then that turns into a fetus, at least in mammals such as yourself. When animals make this zygote and eventually a blastula and a gastrula, a hole forms, and that hole is known as a blastopore. It's known as the first hole. That blastopore is the beginning of the gut or the digestive tract of any animal. In primitive animals, the blastopore turns into the mouth of the animal. The gastrula of a jellyfish has a blastopore, which eventually turns into the jellyfish's mouth. And that mouth is very interesting because its job is to take in food and also to excrete it. The jellyfish has no anus. In more complex animals, when the gastrula forms, then it forms a blastopore and a gut, and then eventually it forms a second opening. And this second opening eventually becomes the anus of the animal. This is true in mollusks, in arthropods, in earthworms, and in other invertebrates. In other animals, such as vertebrates, then the story is different. The blastopore becomes the anus, and the other opening is the one that becomes the mouth. So you see the gastrula makes a blastopore and the other opening. And the fate of the blastopore and the other opening really depends on what kind of animal we're talking about. In echinoderms and vertebrates, the blastopore becomes the anus. And in arthropods and mollusks and worms, then the blastopore becomes the mouth. Those are called protostomes. And in some ways, they're very much the opposite of the deuterostomes.
Okay, so on my notes, the first thing that I would write on my notes is cleavage. Okay, so cleavage, let's do this in a different color, maybe blue. So cleavage is going to be the cell division in a zygote that creates the blastula. The blastula is going to be your hollow sphere of cells, like it's in your embryonic development. And then we come down to these terms. We have protostomes and we have deuterostomes. And so if we kind of back up and we look at this image over here, here is where it is showing us different types of cleavage, protostomes versus deuterostomes. So in the cleavage, when your first cells are forming and going through this mitotic development, right here, cleavage, if it's a protostome, it is a spiral and it's determinate, meaning that those have specific cells they're going to turn into. Now, on a deuterostome, the cleavage is radial, and so they're stacking on top of each other, and it is indeterminate, meaning that those cells can become any other type of cell. So with a protostome, the first opening, as it's showing down here, the first opening is going to be the mouth. So I would note that in my notes. First opening becomes the mouth. In deuterostomes, it's the opposite. The first opening becomes the anus, and then the second opening becomes the mouth. So that really needs to be noted. And this has to do with your digestive tubes and the coelom formation. Okay, so now we are moving on to kind of a different set of notes, and I would possibly set them up. You could set them up like going across, or you can use Roman numerals going down. So in the first column, and right now we're talking about protostomes, and we're talking about invertebrates only. And our first phylum that we're looking at is Porifera. Now, Porifera is going to be our simplest form. I would write that down in my notes, simplest form. Now, an example is going to be sponges, okay? And they lack true tissue. They are asymmetrical. They are protostomes. They have only a few type of cells. This is important right here. They only have a few different types of cells. And they have a sac-like body. They are also sessile filter feeders. So that means that they have these tiny pores or ostium, which are just opening. And so the water and nutrients and tiny little food particles filter through that. And so that captures the nutrients. So these will reproduce asexually by fragmentation or by budding. But basically, I would have they lack true tissue. They are asymmetrical. Protostomes have only a few types of cells, and they're filter feeders. Now, the next phylum that we're looking at is cnidarians. And so your cnidarians are going to be, your examples are going to be jellyfish and hydra. So these, most of the time, they are tubular shaped, tubular animals. This needs to be noted right here. Specialized stinging cells called cnidocyst. This is the second thing I would note right here, is they have a two-layered body sac. So if we look at them right here, we have different kinds of like jellyfish. We have sea anemone, some like coral, Portuguese man-o'-war, and jellyfish. These are all cnidarians. You need to note this, that they lack a brain. They have sensory structures that are around the body that detect and respond to stimuli from all directions. Now, if you notice this, that hydra those are normally going to be your freshwater cnidarians, and their tentacles can respond to stimuli. They reproduce sexually and asexually. They have a nerve net, which is going to be an interconnected nerve cells. They do not have a brain. And then you have your true jellyfish. Now, the next phylum that we're going to move to, the mollusk or mollusca phylum. And so there's over 100,000 different named species. Now, what we need to note about this, here's the first thing, is they have a 3-body part plan. So the first part is they have this visceral mass, they have a mantle, and that mantle is what secretes the shell, or it helps contribute to the development of the gills and the lungs. And then the third thing we need to note is they have a foot that's adapted for locomotion, attachment, or to help capture food. We need to note that they have also an open circulatory system, except for the class of cephalopods, which are mussels, clams, and squids. So in mollusks, the nervous system consists of several ganglia that are connected by nerve cords The true coelom is reduced and largely limited to the region just around the heart. And so in their heart, they pump what is known, it's not blood, but it is known as hemolymph through a vessel called a hemocoel. Now, we looked at also at bivalves, which are going to be your clams, your oysters, and your mussels. And so that is going to be a different class, is how that will be listed. So I would label this as A. Our first class we're looking at is bivalves. And these have a shell, is how I would note it. Or you could just put a hinged 2-part shell. And note that you have clams and mussels in this. They have an open circulatory system. Now, other mollusks, our second class that we can look at is gastropods. Now, this is going to be your largest class of mollusks. Now, examples of this are going to be snails, slugs. Okay, they have an elongated, flattened foot on the bottom, and most of them have that one-piece coiled-up shell and with a well-developed head region. The third class we look at is cephalopods. And these are still, again, in the phylum of Mollusca, but it is a class of cephalopods. In that class is going to be your squid and your octopus. And so they force water out of their mantle cavity for propulsion. They have tentacles and arms. They have a beak that is used to tear prey apart. And they have very well-developed sense organs. Now, the next phyla that we are going to look at is annelids. And so what annelids, the way people most likely remember these is they are known as the segmented worm. They have a specialized digestive tract. You need to note this. They have a closed circulatory system, a ventral solid nerve cord. Most of them are marine, but not all of them. They have a specialized digestive tract in here. I would note that. One type of annelid that we're pretty familiar with is earthworms. And so they have setae that protrude in pairs directly from the surface of the body. You need to note, if you hadn't already noted it in your notes, that all annelids, they have a specialized digestive system. So food is going to be drawn into the mouth by action of a muscular pharynx. And then it's stored in a crop and ground up in a gizzard, kind of like a bird. So they have digestion and absorption occur in a long internal intestine, and waste is going to be eliminated through the anus. Now, earthworms are segmented, and you can notice they'll have the body rings. They have a coelom that's divided. They have branched blood vessels in each segment. Now, earthworms can be hermaphroditic. And so 2 worms, what will happen is they lie parallel to each other, facing in opposite directions. And so they fuse a mid-body segment. They secrete mucus, and it's protecting the sperm from desiccation, which means drying out. And there's no larval stage. Now, with an earthworm, we said that they have like a specialized digestive tract. And so if you'll notice in this picture right here, the coelom, right here, notice how large of a space and an opening that we have here. And then also another thing to note is they have that central nerve cord. Other annelids in that phylum are going to be clamworms, which are predators, leeches, and we find leeches are usually found in freshwater habitats. They have the same body plan as other annelids. But they also have 2 additional suckers. Flatworms are going to be in the phylum of platyhelminths. So platyhelminths, that is your phylum name, and they are known as your flatworms. They have a very flat body, just like the name says. You need to note that they have no specialized circulatory or respiratory structures. And so that means that they have this sac body plan with 1 opening and no body cavity. Now, examples of flatworms are going to be planaria, which planaria look like they have this head, and they have what are known as eye spots. They're not actually eyes. And They have a gastrovascular cavity, and then they have this pharynx extended through their mouth on this part. Now, planarians, they live mainly in fresh water. They have the head is a blunty arrow shaped. They have 3 types of muscle layers. Planarians can reproduce either asexually or sexually. They're hermaphroditic, so they can practice cross-fertilization. They can also reproduce asexually by regeneration. Now, another one is going to be parasitic flatworms. These are also going to be in your platyhelminths. And so we're looking at flukes, and we're also looking at like tapeworms. And so some of these have a head that has been replaced by hooks or suckers for attachment to a host. Tapeworms, they behind their head region is called a scolex. And so it's a long series of proglottids that are found that contain a full set of both male and female sex organs. They have a complicated life cycle, usually involving several different hosts. So if you look at this species of Tinea solium, that is what is known as the pork tapeworm. And that has the human as the primary host and then a pig as a secondary host. So this is just showing us the life cycle that if you eat pork that is raw or undercooked meat, then we become the primary host. We ingest it containing what are known as bladder worms, and then the worms attach to the human intestine where it matures into a tapeworm. And then as the tapeworm grows, the proglottis matures and then eventually fills with eggs. So then those eggs leave the primary host in our feces, which then can contaminate water or other vegetation. And then the cycle starts over again. Then you have hogs or other livestock, cattle, eating those eggs, and we start that whole cycle again with tapeworms. Now, if we look at this term, ecdyzoans, what this means, you need to write this down. This is going to be under our arthropods. That this word means stripping off or shedding is what I would put, to shed or to molt. So these are going to include roundworms and arthropods. Now, if we look at roundworms first, that's going to be a whole separate phylum. That's the phylum of Nematoda. Here, I would write down nematodes are going to be known as your roundworms. We said that they are ecdyzoans. That means they shed or they molt. They contain a pseudocolum, which means a false body cavity that is completely lined by a mesoderm. These are also going to have separate sexes. Now, roundworms, the nematodes, these are going to be the most numerous multicellular organisms on Earth, and they're found in almost every environment. Examples of these are going to be Ascaris, and also Trichinella and your heartworms. And so Ascaris are going to be your intestinal roundworms. And this is kind of creepy that they can lay over 200,000 eggs a day. And trichinosis is from eating the parasite Trichinella, and that is eating rare or undercooked pork is where we get those. We hear of filarial worms quite often. We don't hear this term, but we hear heartworms, and that is what affects our dogs and causes dog fatalities is heartworms. Then there is also the Wucheria, which can cause elephantitis. So these worms use a mosquito as a vector to transmit disease to humans, and then the limbs swell up to a huge monstrous size right here. Here's a good example of it. So this is the worms right here. And then this is how it gets into your tissue, and it creates a cyst into your muscle tissue. This phylum, here's the next phylum, is going to be Arthropoda. And again, this is another one like the roundworms that is ecdyzoan, which means that it has, it sheds or it molts. I would put on my notes that there is an exoskeleton composed of chitin, so it must molt or shed its exterior as it grows. It is segmented, but some segments are fused into regions. So what you need to know is that there's segments. There is going to be a head, a thorax, and an abdomen segment. You also need to note that there is a well-developed nervous system. You have a brain and a ventral cord, and you have eyes that are either simple or compound eyes. Now, examples of arthropods. Here we're showing a dragonfly. We also have crustaceans, which are going to be in there, which are going to be your shrimp, lobster, crayfish. Most of these live in marine and aquatic environments. They have that crusty exoskeleton. There's your crustaceans. Your crayfish is another one. The thorax bears 5 pairs of walking legs. They have separate sexes. We also, in the arthropoda phylum, we have centipedes and millipedes. And the big thing, we also have insects. You need to know that that is an example of an arthropod. And in these body segments, the head bears a sense organ and mouth parts. The thorax has 3 pairs. This is what makes an insect. 3 pairs of legs and sometimes 1 or 2 pairs of wings. The abdomen contains most internal organs, and most insects go through complete metamorphosis. They live on land and breathe through a trachea. So insects are going to be one of our most diverse phyla. So we also include your horseshoe crab, your scorpions, and spiders. Now, moving on to the next is going to be our only deuterostome. All the others that we have talked about, those phylums, those are all going to be protostomes. So I would note that, maybe highlight your porifera, your cnidarians, platyhelminths, nematoda, mollusca, and annelids, and arthropods. Those are all protostomes. Your only invertebrate phylum of deuterostomes is your echinoderms. Okay. Now, I would write this down so that you can help remember this. Echino, right here. This means spiny. I don't think I can write it with this marker right here. But what that means is spiny. And so derm is an outer layer or skin. So spiny skin, spiny outer layer. So we already said they're invertebrates. And they have radial symmetry as adults, bilateral symmetry as larvae. I would note this right here, radial symmetry as adults. They have not an exoskeleton, but they have an endoskeleton. And so it's made of calcium-rich plates, and it has spines. It has a central nerve ring with branches. And then here's the big thing that they also have too that's really noted is they have a water vascular system that helps for locomotion, which means moving. It helps in feeding, gas exchange, and sensory reception. So our examples of echinoderms, so our spiny skins right here, those are going to be like our sea stars. And you notice here it's like radial symmetry. Your sea apples, your sea urchins, your sand dollars are also an example of them.